Perfect. Thank you. Okay, well, my name is Marta Jarzyna, and I'm a communications director and the board member for the International Biogeography Society. And it is my immense pleasure to introduce Dr. André Bellvé, who will be speaking to us today. André is currently a postdoc at at Ohio State University in Columbus, Ohio, in the Department of Evolution, Ecology, and Organismal Biology. So he is very closely positioned in terms of his office to my own. I can always ask him questions if I have any questions. But because of he's being employed by the Ohio State University, I have had a good opportunity to learn more about Andre's research, and I found it really fascinating, and I thought it would be a great opportunity for him to speak to our society and to our members and to share his really, really interesting research results. Dr. Bellvé obtained his bachelor's, master's, and his PhD degrees, all from the School of Environment at the University of Auckland in New Zealand. So he's coming to the US from literally the other side of the world. And his research primarily focuses on paleoecology and particularly ecosystem level changes over the past 21,000 years. And he's interested in how data can be effectively utilized to gain more robust insights into community assembly and into ecosystem functions. So today he will share with us his interesting research on how we can reconstruct some of the ecosystem functions from all kinds of interesting data. So without further ado, I welcome Andre, and please, you can share your slides now and jump right in.
Kia ora. Thank you, Marta. Uh, can everybody see my screen? Is that sharing correctly?
Yes.
Perfect. Well, tena koutou, tena koutou, tena koutou katoa. Welcome, welcome, welcome. Thank you for coming along to my talk. And thank you to the IBS for the opportunity to talk about my research and some subjects that are quite close to my heart, being a seabird lover. So this is work that I conducted with George Perry, Janet Wilmshurst, and Jamie Wood primarily, although there's a lot of collaborators because there's quite a few projects tied into one that I'm presenting. It was all conducted at the University of Auckland with joint funding from the Manaki Whenua Land Care Research Group. Uh, so I'd like to start off by just first saying a quick thank you and acknowledgement to the Tangata Whenua, the people of the land of Aotearoa, New Zealand, who are kaitiaki or guardians of the seabird species I'm going to be talking about today, and without that guardianship, most of these species would not still exist, as we'll get into a little further a little later. I also need to say thank you to a bunch of different people, but first and foremost is Eden Whitehead. She's another scholar from New Zealand, and all of the photos that you see in my presentation are hers, unless they're explicitly referenced otherwise. So you should go check out her photography. She's quite a phenomenal wildlife photographer. I'd like to thank a whole bunch of people for all the support and data they provided. Most of these people or actually all of these people are co-authors on various projects. That are still in the works, and the CBB, UOA, and the School of Environment for various pools of funding that I received to complete this research. So without further ado, I guess I'll make a case for seabirds as important vectors of nutrients from land to sea. And I think Macaulay, a paper by Macaulay and colleagues in 2012, characterized this really perfectly. They compared two islands, one without seabirds and one with seabirds. And they found that when seabirds were present in an island, they fertilized the soil, which led to greater primary productivity, diversity of vegetation, and just more of it. And coupled with that increase in terrestrial vegetation, there was large amounts of foliage or greater levels of foliage being blown into the nearshore coastal environment alongside runoff from soil or runoff from the nutrient-rich soils into the coastal environment as well. And that promoted phytoplankton and plankton diversity and abundance, which then attracted things like manta ray. So the simple presence of things like seabirds, which derive their nutrients from the marine environment, in terrestrial sites can form these really long chain ecological linkages that promote biodiversity, increase abundance, make ecosystems more resilient, and can improve their function in terms of carbon sequestration or various other things. And some work by Chris Doherty in 2016 showed that there's even longer chains to these links. Things like cetaceans move nutrients from the seabed, which is extremely rich in phosphorus and nitrogen, and introduce it to the pelagic sphere. And subsequently, seabirds and Andromous fish, sorry, move these nutrients inland, where they're then dispersed by things like bears or megafauna. And in his work, he showed that these nutrient fluxes have been reduced quite catastrophically. So 70% reduction in cetacean nutrient fluxes and well over 90% being moved inland by Andromous fish and terrestrial herbivores. But you'll note that seabirds weren't, they never reproduced what seabirds did in the past here. But these nutrient fluxes are really important, especially in New Zealand, which is really what I'm going to be talking about today, because there's very few sources of bioavailable phosphorus on land. So a huge portion of this comes from marine environments. And we know from Doherty's research that Aotearoa, New Zealand, and Oceania in general are hotspots for seabird nutrient fluxes. Seabirds are a key importer of these nutrients in these areas. And the reason for this is that New Zealand has often been called the seabird capital of the world, and for very good reason. So there's approximately 360 species of seabird globally, and roughly 25% of them have breeding grounds in New Zealand, and 10% of seabird species are endemic breeders being found nowhere else. So in Aotearoa, New Zealand, we have a really disproportionate share of seabird species. Now, unfortunately, 90% of them are threatened with extinction. And that's due to a bunch of different sources, but pretty much all of them anthropogenic in one way or another. So it's things like habitat destruction, clearance for houses, the introduction of mammalian predators, which we never had in New Zealand prior to the arrival of humans. So things like mustelids, cats, rats, stoats, pigs, dogs, etc. The pollution. So on the right-hand side here, apologies for the graphic image, but this is a shag that was caught on some discarded fishing line and was unfortunately still alive when this image was captured, and we couldn't do anything to help it. But these seabirds are unfortunately getting caught up in human pollution and then bycatch from fisheries. Now, not all seabirds are affected equally. Some research by Richards in 2021 showed that biological traits of seabirds can help predict their extinction risk and vulnerability to anthropogenic threats. And you can see that there's some groups, things like charadriforms, which are gulls and terns, and suliforms, are relatively unaffected or relatively low risk and not threatened so much by human impacts, but that procelariforms, the tube-nosed seabirds, are disproportionately covered by or affected by anthropogenic risks. So procelariforms, or the tube-nosed seabirds, as I said, includes things like albatross, petrel, shearwaters, prions, and storm petrels. It's a very diverse group of seabirds, and they're really true seabirds in that all these species obtain all their nutrients essentially from the marine environment. They spend very, very, very little time on land. They feed out at sea on plankton, squid, and fish, and they really only return to land to breed in large aggregate colonies. Now, the ones I'm going to be focusing on today are the burrowing procelariforms. So these are species that are typically less than about 1,600 grams. And in New Zealand, the burrowing procelariforms represent roughly 50% of the seabird species richness. So it's a considerable diversity of the seabirds we have. So that kind of gets me to my research and what we wanted to do, which is reconstruct nutrient fluxes by burrowing procelariforms in Aotearoa. And to do this, we needed to know three key things: where they used to breed, what nutrients they flux, and how that flux has changed through time. So we'll start with the first question as I break this apart into pieces, which we tackled with ecological niche modeling. So if you're not familiar with this, in its most simplistic form, we start with some environmental predictors. So this might be things like rainfall or elevation or temperature. Combine it with some presence data of the species of interest, funnel it through a algorithm, and we can make associations between these presences and environmental predictors to understand where the species is distributed and create these nice heat maps. So to figure out where they bred, we first needed some presence data. And we got this from three primary sources. First is Holocene aged fossils. New Zealand has a really rich natural archive of fossils, and due to a relatively short history of human occupation, humans have only been around the last 800 years or so. We have a really rich archive of where seabirds used to be. And we combined this with historic records. So that included things like historic accounts from naturalists like Andreas Reichek, pictured here, and old newspapers from when colonists arrived in New Zealand around 1840, where they noted where they found petrels and of various species across all of New Zealand. And the final source of data was modern surveys. So that's really where they are today. So it gives us a look at three kind of unique time points and also three unique record sources in terms of the biases and what kind of habitats they might show. We took these presence data and we broke them into size classes. 30 to 200 gram seabirds, 200 to 600 gram, and 600 to 1600 grams. And I'll get more into exactly how we did this because it wasn't just simple body mass breaks in a second. But we took these presence data and these size classes, and for each size class, we fitted ecological niche models using mixed principal component analysis. This is a PCA where you mix both categorical and continuous data. And maximum entropy modeling, which is a type of machine learning for creating distributions particularly. So for the size classes, what we first did was take a bunch of traits in terms of both morphological things that might affect their burrowing preferences or their colony preferences, things like wing length and other morphological traits that relate to their ability to burrow or to disperse inland, things like Kip's distance, as well as their nesting ground preferences from published data. And then we clustered them. And what you can see here is kind of three clear groupings of seabirds. And these really, fortunately, fall along body mass breaks. Body mass was not one of our traits, though. So in this bottom group here, we have our 30 to 200 gram birds, our 200 to 600 gram birds, and our 600 to 1600 gram birds. There's a little bit of an overlap here, but for the most part, they fell along these lines quite nicely. So I'm going to be referring to them by these size classes for the rest of this presentation. And what we did was compare different record sources. So these are the results of our mixed PCA. And the gray points are a sample of background points from New Zealand's environments. And we use that to calibrate the PCA, and then we plotted our points and kernel density estimates around different record sources. And hopefully what you can see, this is the 200 to 600 gram seabirds, is that the historic and fossil records, historic in pinky purple and fossil in yellow, captured very different portions of New Zealand's environments. There's more inland and high elevation sites in our historic records, while the fossil records were closer to the coast, more lowland, and occupying portions of the niche that weren't captured by historic records. And when we compare this with the combined historic and fossil records shown here in orange to the post-1990 records, you can see that for this size class at least, there's been a very stark contraction. We've lost most of these inland sites and high elevation sites. With the present day, distribution being much closer to the coast and occupying only a small subset of where they previously existed. So this kind of novel synthesis of historic and fossil records helped us quantify and account for biases in each of these record sources and give us a unique perspective of the species or size class's ecological niche, and shows some evidence, at least in this environmental space, of range loss. And if we go over to our maximum entropy models of the same data, what we found for each size class was evidence of range contractions. So to read these maps, warm colors indicate a higher probability of presence. And while faint, what you can see is for this 30 to 200 gram seabird, they were present much further inland than they are today, which is shown here on the right. There's no warm colors, are really just relegated to offshore islands, which often act as predator-free sanctuaries for these birds. So we've had a mainland extirpation for these. And this is much starker for the 200 to 600 gram birds, where they are present very far inland, all along the southern Alps here, on the west coast of the South Island, all around the volcanic plateau in the North Island, and now they're all but gone. And we see much again the same for the 600 to 1600 gram birds, this mainland extirpation and a relegation to offshore islands and the coasts. So across all three size classes, we saw this evidence of extirpation and a loss of seabirds. And if we go back to historic accounts, they can provide a lot of context for why these extirpations likely occurred. So this is Andreas Reichek, who I mentioned earlier. He noted in 1885 about black petrels, or Procellaria westlandica, that as I examined the burrow, and he's talking about black petrel burrows here that he found, as I examined the burrow, and on putting my hand in, it was severely bitten. So I tried again to take the egg from under it. I was again severely bitten. My dog went to the bird and attacked him furiously. He remarked on the same page in another paragraph, these birds, which were very numerous on the Little Barrier Island, had become very scarce, but I found the remains of many which pigs and dogs had destroyed, which I found very sadly ironic, that he caused the death of many of these birds and then remarked on their cause for decline. And a contemporary, Oliver in 1890, mentioned something to much the same effect, that the introduction of rats, cats, stoats, weasels, ferrets, and hedgehogs has greatly reduced their numbers. So these historic accounts really give us perspective on what drove these populations extinct very quickly. And I guess in kind of summary for this bit, what we found was that combining record sources can better represent ecological niches because they overcome the biases inherent in individual record sources, and that reconstructing distributions with these can help us uncover histories of extirpation, and that historical accounts can provide context for these declines. So that kind of wrapped up where they bred, and next we need to figure out what nutrients do seabirds flux. So for those who haven't worked in nutrient flux space before, there's two primary methods that research often uses to figure out how much nutrients are being fluxed. The first is direct estimates, and it's kind of what it sounds like. You collect and measure a bunch of excreta from the taxa of interest. You work out the concentration of elements in that excreta, and you figure out the quantity of excreta that an individual produces on a typical day. And unfortunately, this method is often very labor intensive. You have to have a lot of time in the field to collect the samples. And it can be very challenging to collect, as I know from firsthand experience. Surprisingly, seabirds are not always very happy to give up the goods. So that has led a lot of people to indirect measurements of seabird nutrient fluxes. And this is often things like bioenergetic models to estimate excretion rates. And they use things like metabolic rate allometry, so using body mass to figure out correlations with the energy needs of an organism, the energy content of a species' diet, the nutrient content of their diet, and assimilation efficiency of the nutrient intake for their metabolic needs. And from that, people estimate how much nutrients are fluxed. But there's many unknowns when we use these models because they're not based on direct estimates, but rather basically a formula of metabolic rates relationship with dietary intake. So there's a lot of environments and species dynamics that aren't accounted for. So for this project, we wanted to figure out global seabird nutrient fluxes presently. So this is kind of ignoring past dynamics. They'll come back in later. And a paper published by Otero and colleagues back in 2018 estimated that seabirds flux approximately 591 gigagrams of nitrogen. That's 591 million kgs of nitrogen annually and 99 gigagrams of phosphorus, which are phenomenally large numbers. And they base these estimates on bioenergetic models. Now, I'm going to take a second to go through this equation and break it down because we used a different method to try and get estimates of nitrogen and phosphorus ourselves. Now, this right-hand side of the equation here is really just accounting for the population size of a given bird and its time. And we used exactly the same data for these. So this part of the equation, in effect, for comparing my results with, or our results with Otero's was a constant. So I'm just going to gray that out. This other chunk of the model, though, is really where the assumptions are being made. And I want to break them down because I think it'll help contrast the results I found, or we found, with that of Otero's. And this top portion, the numerator of this division portion here, basically says that metabolic relationship with body mass are constant or scales constantly across all taxa. So that means the metabolic rate of a small penguin to a medium-sized one would be the same of an equally sized, say, albatross to a larger seabird or something, or a small petrel or something to a larger albatross. So it's a constant across all taxa. This FEC component assumes that the energy content of a seabird's diet is based entirely on fish. So all seabird eat fish, which is, again, quite a big assumption. Fish are quite energy dense as far as nutrients go. But we know that a lot of seabirds eat squid and krill and other plankton and the such. So this bit might be a bit of a stretch. This AF is the assimilation efficiency constant, and this was held constant for all seabirds and was based on assimilation efficiency of macaroni penguins. So that means that they were assuming that petrels, gulls, terns, cormorants all have the same exact assimilation efficiency of their food as macaroni penguins. And then finally, this FNPC is the concentration of nitrogen and phosphorus in a seabird's diet, and they base this on fish, a fish diet, which again might not reflect the diversity of diets that a lot of seabirds have. So what we did in contrast to this was collect a whole bunch of fresh excreta from all four major orders of seabirds. That's the Charadriformes, Suliformes, Spheniciformes, and Procelariformes, which I'll go through in more detail in a second. And combine and analyze this with a CH&S elementar, which gives us measures of carbon, hydrogen, nitrogen, sulfur, as well as an ICP-MS, or a inductively coupled plasma mass spectrometer, which gives us estimates of everything basically heavier than sulfur for our intents and purposes. So we combined our direct measurements and the collection of fresh excreta that we had with published data on nutrient content or element content and concentrations in seabird feces. And what we found is that there are very stark order-specific differences amongst all seabird species. So this is a measurement of fresh excreta. And you can see that things like Procelariformes or tube-nose seabirds have the highest nitrogen content, but the lowest phosphorus content. Whereas Suliformes, things like cormorants and boobies, and gannets, have low nitrogen, but very high phosphorus estimates. And that penguins, or Spheniciformes and Charadriformes, things like gulls and terns, are sitting somewhere more in the middle. But there's very clear order-specific trends amongst these in terms of nitrogen and phosphorus concentration in seabird feces. So that gave us a concentration, but we still need to figure out how much birds produce. So to do this, we went back to the literature and gathered up a bunch of published estimates of defecation rates for different seabird species and fitted models individually for each of 3 seabird orders against mean adult body mass and the feces produced. Now, the keen-eyed among you will note that the suliforms are missing, and that's because there's no data on suliform defecation rates, so we simply didn't have access to that information. But hopefully what you can see is that there was quite tight and consistent order-specific allometric relationships between body mass and feces production. So with this, we can estimate or estimate some of the feces production for species that we don't have direct estimates on. So we took our models and incorporated the errors from both the concentrations of elements and the allometry of feces production to create uncertainty bounds around our estimates. So what you can see here is for each of these 3 orders, we have the quantity of nutrients that get fluxed onto land, shown by this solid black line, and the total quantity of, say, excreta in this first column, nitrogen in the second one, and phosphorus in this third. And what we estimate is that these 3 orders together flux approximately 530 gigagrams of excreta. That's not nitrogen, so don't get that confused with the previous estimate. And that's equal to about 48,000 school buses worth of poo every year, which is a pretty huge amount, and quite ecologically important for a lot of vegetation communities in particular. And of this, approximately 61 gigagrams, so 61 million kgs of nitrogen. And that's about 5,500 school buses. And finally, 13.8 gigagrams of phosphorus, or 1,200 school buses. And if we compare these to the previous results of nutrient fluxes by seabirds, they're significantly lower. So shown here in the top right-hand corner in red are the previous estimates, and they range from about 2 times. The previous estimate was 19 gigagrams. We found about half that. And for phosphorus for procelariforms, our estimate was 50 times lower than previous estimates. While these numbers that we found are still huge and obviously extremely important for a lot of vegetation communities, they're orders of magnitude lower than previous estimates. And we hypothesize that this is probably because we accounted for a lot of differences in seabird ecology, as well as incorporating a bunch of uncertainties we had in our estimates. So by making order-specific models rather than relying on, say, the assimilation efficiency of penguins to represent all seabirds, this can drastically change what we estimate that seabirds flux. And what we present here is not only revised estimates of seabird nutrient fluxes, but also hopefully a method for scaling from direct measurements, which are arguably much more reliable because we're measuring exactly the thing that we're interested in and scaling them to limit, I guess, the amount of primary data we still need to collect, because it is extremely expensive and time consuming and often just impossible to get. So that gave us what nutrients they flux. And I'll come back to how we did that for New Zealand a little later. But the next thing we wanted to understand was how these nutrient fluxes have changed through time. So we know that procelariforms collapsed very quickly after the arrival of Europeans in New Zealand in particular, and the introduction of mammalian predators. But we don't have any empirical estimates on how the population collapsed or what the population looked like before the mammals and humans arrived. What we do have is kind of rough accounts from both Reischek and Oliver that it appeared that the seabird population collapsed roughly within 30 to 50 years of mammalian predator introduction. But what we don't know past just population estimates is how the populations were interacting with one another during these declines, because seabirds show very strong intraspecific attraction to breeding grounds. They have very high rates of philopatry. That means that they come back to their natal breeding ground when they're sexually mature and ready to reproduce themselves. So to gain insight into the dynamics of how quickly seabird populations collapsed and what impact the arrival of predators had on one population to another, we developed simulation models of individual seabird behaviors to see how these play out and interact. So we know that seabird colonies are not static and that they are linked through processes like immigration and emigration. But they're also spatially explicit, meaning that they occupy very defined breeding grounds, but are also temporally dynamic, right? They can fluctuate through time. They're going to respond to environmental conditions and the populations of other colonies, right? If birds are emigrating and immigrating. So we used agent-based models, or ABMs, to try and untangle some of these population dynamics and understand the sources of mortality in seabirds. And to give you a high-level overview of how our agent-based model worked, we started by going to the seabird literature and getting empirical estimates for everything we possibly could. Our model, or our model system, starts with some starting population and parameters for an island. That's like how many birds it can hold, how densely aggregated the habitat is, etc. And once we have these initial populations, it immediately goes to a round of recruitment. So birds age a year. Those who are sexually mature are then going to enter the adult breeding population and choose an island for their breeding grounds. So that's a philopatry check. Some birds might immigrate between islands. Once everybody's chosen an island, we then establish males on the island, and females find a mate amongst these males. Those who are successful will then hatch and fledge chicks. Some mortality gets introduced, so this can come from various sources, i.e. like natural sources, which is the mortality without the presence of mammalian predators, or predation mortality, which we're attributing to mammalian predators. So that, to be clear, obviously there's predation in natural systems, and that's kind of lumped in with natural mortality, right? That's things that would have predated on seabirds prior to the arrival of humans and mammalian predators. Whereas predation mortality we're explicitly defining as the more added mortality that's attributed to the presence of things like cats, rats, and stoats. And because this is a agent-based model that's developed in silico, we have perfect knowledge of the system. So we claim a census. We figure out the number of birds settling on each island, how many are hatched and fledged, what mortality, etc. And then a new year begins and the whole cycle starts again. And we can repeat this for many different scenarios and rerun this over many iterations, because all of these have dice rolls and are essentially stochastic to some degree. So this is what the front end of the model we developed looks like. And I don't want you to try and read everything. I know it's pretty overwhelming. There's a lot. But what I wanted to show you is that we have controls for things like chick, juvenile, and adult mortality, old age mortality, how many seabirds are recruited, what age they first breed at, philopatry rates, predation rates. So again, this is mammalian predation rates, the impact of El Nino Southern Oscillation cycles on breeding, because we know that they can depress reproduction in many seabirds populations, controls for emigration, controls for the habitat, how quickly burrows are prospected, how quickly they collapse. So what we started with was basing all of these parameters on empirical estimates for moderately sized petrel. So we empirically grounded all of the estimates that we could, but models like this provide us a lot of flexibility in terms of the scenario parameters we set up and the species traits that we use. So this can be extended to other species or other questions. And it provides a counterfactual platform for a question-based approach. And what that means is we can set up impossible or unrealistic scenarios to see how that changes the results or impacts on the system. So for our ends here, in terms of reconstructing nutrient fluxes, we wanted to understand the impact of predation and ENSO on seabird populations and how they interacted between populations. So we have a two-island system. One island will have predators arrive at this red bar about 50 years into the simulation, and that's the red island. The blue island will never have predators, but is linked to the red island by immigration. The dashed orange line here shows the virgin or the kind of null biomass or number of breeding pairs in this scenario. In this scenario, we just have ENSO. There's no predators arriving, so this red line just indicates when they would, but there's none on either island in this scenario, right? It's just to get us an estimate of how many seabirds arrive on the island, what the population unimpacted by predators might look like. It's kind of like a real world scenario prior to the arrival of Europeans. So if we run the same exact simulation, but add predation at the 50-year mark, what you can see is both populations plummet. So to reiterate, the predators are only arriving on this red island, which crashes within 50 years and is extirpated probably within about 70 years. And we see the total collapse of the population. But even on the island where no predators arrive, we get a suppression of the breeding population by more than half. So what this suggests is that things like immigration can create source sink dynamics between populations when there's different sources or different levels of mortality in two populations, particularly because these species are so gregarious and tightly linked. So this scenario had predation and ENSO. That's basically our real world scenario for today. Now we set up a third one, which was counterfactual, and that's that we drop ENSO. So there's no climate impact, which we know obviously there is in the real world, but we wanted to understand what the added effect of ENSO might do. And we found that in the presence of just predation, no ENSO cycle, breeding mortality, the populations were still suppressed by the arrival of predators, but they no longer tipped into negative growth and collapsed completely. And while this might seem like kind of a silly scenario to run, what we know with climate change is that there's going to be more pronounced ENSO effects, and that phases like La Nina and El Nino are going to have stronger lock-in. So they're going to be persistent for longer periods of time, and the effects are going to be more pronounced with things like marine heat waves, which quash prey availability and prevent seabirds from breeding, or just they starve to get to death altogether. So what we really wanted to gain from this is that understanding of how important ENSO cycles are. And what it means is that populations that might currently be holding on, if climate change makes breeding more difficult for them, it could tumble them into complete collapse. So altogether, these simulations help us characterize unobserved declines, untangle sources of mortality and population dynamics, and help us understand if what we see today is representative of the past or what might happen in the future. So I mentioned early on, the real question was how we can reconstruct ecosystem functions in paleo environments. So we needed to tie all this research together. And what we did is take the distribution models of where things are in the past and present to figure out where they are. And then the nutrient flux estimation to figure out what is being moved. And then finally, the colony dynamic simulations to figure out the timing and characterize the collapse and decline of these seabird populations. So those who are familiar with E&Ms will know that they produce maps of probability of occurrence. So past studies like Chris Doherty's to estimate seabird population sizes have just scaled density allometrically across all avifauna. We had the luxury of working in a much smaller system with a much more clearly defined or a much more specific seabird order. So we thresholded the probability of occurrence at 51%. So only 51% or higher was accounted for. And that's basically indicating a probability that the species is more likely to be there than not. We went to the empirical literature and found estimates for the minimum known density that seabirds nest at and the median density that seabirds nest at. We didn't want to go for max because that can be really skewed by outliers. And then we linearly scaled from 51% occurrence probability up to 100% occurrence probability from the minimum density of minimum breeding density and the median breeding density for each of our three size classes to create maps of seabird abundance in the past and present, and then translated this into nutrient fluxes using our allometric models and nutrient or element makeup of feces. And what we found is that in New Zealand today, approximately 8% of the former area is still receiving marine nutrients. And that's about 57,000 kilometers squared, or roughly 7.8 million soccer fields. And today, it's just 4,500 kilometers squared, all pretty much relegated on the coast. So there's only 8% of the area, but it's more concentrated. And what we found is the total flux volume is about 17% of its former size. So this dashed orange line here is the present, while the purple line is the past. And hopefully what you can see pretty clearly is this orange line collapses very quickly, with 99% of the nutrient fluxes by burrowing prosolariiforms arriving within 1 kilometer of the sea shore. While in the past, we estimate that roughly 25% of the flux arrived more than 15 kilometers from the shoreline. So the nutrients were going much further inland, which is really important because that means there's more systems that they're going to pass through before they eventually return to the ocean. So more vegetation, more ecological communities can benefit from the nutrients. And to put some numbers to it, what we estimated for all three size classes together is that in the past they fluxed about 6.2 gigagrams of poo, whereas now it's less than a quarter of that size. And that was about 1.6 gigagrams of nitrogen and 0.11 gigagrams of phosphorus, or 110,000 kilograms of phosphorus every single year, which are still pretty phenomenal numbers, particularly considering how big New Zealand is. So to tie in our timing of decline, we conducted 3 agent-based simulations using our real-world maps of seabird populations for each of these 3 size classes on two colonies, one where predators never arrived, i.e. sanctuaries, and mainland populations. And what we found is that for the 30 to 200 gram size class, the population collapsed within roughly 27 years, and its total population was suppressed to about 56% of its size. For the 200 and 600 gram size class, they took longer to collapse, about 38 years, and it took collapsed to about 40% of the population size, while the 600 to 1600 gram birds took the longest to collapse, 56 years, but was only a third of its size today. And what's nice is these numbers line up really closely with empirical estimates that were given with the estimates that were given by Reichek and Oliver of about 30 to 50 years for seabird populations to collapse, giving us a lot of confidence in our results and a loose kind of ground truthing as close as we can get. So there's lots of implications of this nutrient flux loss. Obviously, there's going to be decrease in primary productivity due to the loss of these critical nutrients for plants. And we've clearly altered a lot of ecological communities by letting these species go extinct or extirpated, and broken many likely long chain interactions in the process. And unfortunately, the long-term effects of this are still really unknown as ecosystems respond on the order of decades to centuries. So hopefully what I've kind of demonstrated is that we can reconstruct distributions using multiple record sources. So this can be translated to other lines of research, right? Other functions that animals perform, and understanding past and present distributions. And that things like elemental analysis and allometry can help us scale estimates that are otherwise too laborious to collect. If we select our samples well, we can make a lot more use of the data that we have. And that finally, simulations can help us untangle metapopulation dynamics to show that the present may not be analogous to the past and help us understand the timing of these declines to figure out how rapidly they collapsed. And that can help us understand the long-term trajectories of these losses. So where to next? I think there's some really interesting work on the horizon in creating temporal isoscapes by basically coring things like trees and sediments and stuff to figure out nutrient signatures of different species, and that can help us refine distributions. There's some work by Matthew Duda that I find really encouraging of using diatoms from lakes to figure out past population sizes and gain more insight into past dynamics. And if we combine the results of models like the one that we have built and I showed here, we can get better ideas of how vegetation dynamics respond. So incorporating vegetation ABMs to understand how vegetation communities respond to the loss of nutrients, to predict what's going to happen in the future, and to try and understand what we're seeing today. And obviously extending this framework to other taxa and functions, right? There's plenty of other species and groups or systems we could explore to understand what functional or ecological functions we've lost through the last few thousand years where human activity has really changed the face of the globe. So with that, thank you again for taking the time to attend my talk, and thank you to the IBS for the opportunity to give a lecture in the Funk Lecture Series. I feel really privileged to do it. I'm happy to take any questions if anybody has any.
Thank you so much, Andre. This was really, really interesting. If you have any questions, please put them in the Q&A box, and I will read them off for Andre. And we have 5 or so minutes for questions, so so there's definitely time. I can kick us off, I guess. I'm just wondering whether you can speculate or whether you see the opportunity to use your results to somehow guide conservation, maybe restoration as well.
Yeah.
In New Zealand, so much is, there's so much literature now on rewilding and how to go about that, particularly in Europe, but also in other places. So I'm curious whether there's any discussion about that in New Zealand and whether you see your research particularly well fitting in terms of guiding that discussion.
Yeah, absolutely. So, I mean, with the stuff that I presented here today, the distribution work, it makes a really clear case for where potential translocation sites might be. And for those who are attending my talk, I swear to God that Marta is not a plant, but I do have a couple slides that talk to this. So using those simulation results, we also explored recolonization. So this is a little bit different and it's very preliminary. So, take it with a grain of salt. But we had our 2-island system, right? The blue island starts off uncolonized, no seabirds present, while the orange island is colonized. And we want to understand how quickly seabirds might recolonize sites. And if we have a situation where there's 0% mammalian predation, what we found is that it could take upwards of 100 years for colonies to naturally reestablish on their own. And if we have any added chick predation, like there's any mammalian predators left, even at very low estimates of predation, 10%, it's really more in the order of 40%. There's no natural recolonization. So we have to get rid of mammalian predators. And even then, it could take 100 years if we just leave it on its own. Fortunately, there's a lot of really fantastic research and conservation managers that are already trying to restore past populations. And they use a diversity of lures to bring seabirds back, things like visual lures. These are 2 concrete painted gannets and Nigel no-mates. It's a very sad story if you look up Nigel no-mates in the background who fell in love with some of these concrete statues. Acoustic lures where they play callbacks of seabird breeding populations to try and simulate colonies before there are any, translocations to take advantage of seabird behavior, things like philopatry to trick them into thinking that this is their natal breeding ground and get them to return there to stimulate a new population. Obviously, habitat restoration, and then most importantly, predator control. Um, so yeah, thank you for asking that.
Yeah, I did not. Yes, it was not a planted question. I was just really curious, and it was good to see that you have some slides about this.
Yeah, I've
So
had it before, so
yes, I'm sure people ask about this all the time. So given that um, those management strategies are all good, but have you seen any success so far? Is there a success story there at all that you can
yeah,
point to and say this works and and this doesn't?
yeah, it's a great question. Honestly, the success has been quite mixed. So, New Zealand Department of Conservation has been doing translocations since 1995 or so, I think, if I recall my facts correctly. It started on Maud Island, and initially there was a really quite quick uptake of seabirds coming back and starting to reuse that colony. This is based on translocations. Uh, and then it kind of tapered off really quickly. So clearly, there's some dynamics that we're not understanding. And what a lot of people have hypothesized is that because there's other big colonies in between, seabirds are getting kind of siphoned off to those big ones when they're coming back. So clearly, there's a lot more to understand. Other islands have had a lot more success. So Great Barrier Island is a fantastic example, or sorry, Little Barrier Island, Hoturu, which is in the Gulf of the Hauraki Gulf near where I'm from, um, and it had predators on it, pigs and rats and stoats and cats and stuff. And it was the one that Reichek remarked on the black petrel populations collapsing. That was declared a predator-free sanctuary after huge effort to eliminate predators from it. And since then, it's seen explosions in the populations of seabirds. So clearly, it's having a massive impact. If we can get predators off and make safe refuge for these species, they can largely take care of themselves. Climate change is a big unknown, I think, but clearly one of the most pressing and immediate concerns is just the removal of predators, and that can have a huge impact. Seabird islands without them tend to explode and are just raucous with life.
Yeah, that makes a lot of sense. So I do not see any questions in the Q&A. Um, I guess we can then wrap up, unless people still have. Let's give folks maybe a couple seconds to put some questions in there. Oh, there it is from Wolfgang Trailer. Thanks for the talk. Which role does anthropogenic nutrient flux play in this context? Phosphorus
Mm.
or nitrogen agriculture?
That's a great question. So, if I understand correctly, and correct me if I'm wrong, you're asking about the role of nitrogen, the movement of nitrogen and phosphorus to land by humans.
Hmm.
So, what we know is that it's going to very different places, right? These seabirds nest on the top of mountains often or big scarps in forests. The picture that's the background right now is taken from an island, Poor Knights Island of Ferry Prion, I think, taking off. Those anthropogenic nutrients never make it there, right? Nobody's dropping fertilizer on those islands. It's all going to agricultural areas, and those are often fields. Um, which means that the nutrients have a are being introduced into environments that are really just used for human food production, and they have very quick runoff because they dump huge amounts of fertilizer on the ground. It goes straight into the rivers, which can eutrophy a lot of freshwater environments and have horrible impacts on them. Whereas the nutrients that seabirds move, they burrow beneath, I mean, at least the ones I was studying, they burrow beneath trees in the middle of forests. So they're delivering nutrients that are getting pretty much immediately uptaken by the forests themselves, and they have a much slower release into the nearshore environments or into other ecosystems around them. So I think the two serve very fundamentally different purposes because they're arriving in different places and they're being transported in different ways, right? So yeah, it's a great question. Humans have modified all kinds of nutrient fluxes in pretty astounding ways, and there's some fantastic research that just came out by a colleague of mine, Andrew Abraham, on how humans have actually modified bird avian nutrient fluxes through bird feeding and showed that the phosphorus and nitrogen that humans are effectively putting into the environment through bird feeding in the UK is astounding and can have huge repercussions on the ecosystems that they are putting them into, which are essentially just urban and kind of rural rural areas. So yeah, I think there's a really interesting intersection between anthropic and animal vectored nutrients.
Great. The difference between runoff and location makes sense, says Wolfgang. So, yeah,
Perfect. Thanks for the question.
yeah. We're out of time anyway at this point. So, please help me thank Dr. Bellvé for this really interesting talk. And it was recorded, so you'll have a chance to take a look at it and listen to it again on our YouTube channel. Or if you know someone who's interested in this topic, please feel free to send them our way. It should be posted within several days. Thank you very much, and we hope to see you for our April FUNK lecture. We will welcome Dr. Lydia Boudreau from Michigan State University then. Thank you, André, and thank you everyone for coming. Bye.
Thanks. Bye.